Alright, welcome back to MCAT Grind. This is the big chapter starting on carbohydrate metabolism. And we know glycolysis and the citric acid cycle are big parts of the MCAT and people dread memorizing it. I dread memorizing it. But here we begin with the first part, which includes glycolysis, the glycogenolysis, glycogenesis, and gluconeogenesis. So to start, we have the transporters, GLUT2 and GLUT4. GLUT2 is found in the liver and pancreas, and their function is to stimulate insulin and to serve as a low detector for blood glucose. Now you'll notice that they have low affinity for glucose, so you need a very, very high amount of glucose to trigger them in your blood. GLUT4 transporter are found in the fat and the muscle tissues, and their purpose is just attachments and to bring the blood glucose into the cells. And they are stimulated by the insulin. So more insulin takes in more GLUT4 transporters onto the surface of the membrane of the cells to bring more glucose in. So GLUT2 is increased by first order kinetics, whereas GLUT4 is zero order. So the more GLUT4 is just in a linear progression line, where GLUT2 you need a lot more concentration of glucose to use to detect GLUT2 efficiently. Now, getting right into glycolysis, we start with the glucose molecule. The glucose molecule, the tally result ends up being 2 pyruvate, 2 ATP, and 2 NADH. So you will see that the bottom half here, when it gets into pyruvate, it does it twice. So starting with glucose, it uses hexokinase or glucokinase, depends on where it is. The hexokinase is in trapping glucose in your muscles and your tissues, whereas glucokinase is only in your liver. And what they do is they trap it in the liver and they sense, and in conjunction with GLUT2 to sense the blood glucose level and to induce later on bringing in of the glucose into the tissues. Now once hexokinase and glucokinase has trapped the glucose, it is converted into G6P, and then isomerase turns it into fructose 6-phosphate. Fructose 6-phosphate then can be used by the enzyme phosphofructokinase 1 into fructose 1,6-bisphosphate. Now you will notice that phosphofructokinase adds a phosphate to it. So fructose 6-phosphate, 1,6-phosphate has another one of a phosphate molecule. So that helps you remember. Phosphofructokinase 2 also brings F6P into fructose 2,6-phosphate. And what that does is this serves as a little secondary mechanism to induce phosphofructokinase 1 to make fructose 1,6-phosphate. So it's like a secondary help to override the effects of the reversal discussed later on. Now, fructose 1,6-bisphosphate now can be broken down into glyceraldehyde 3-phosphate and DHAP. Now, what this does is from a 6 carbon chain, it being cut into 2. So this can go on to make twice 2 pyruvates. In this DHAP pathway, it uses glycerol 3-phosphate dehydrogenase to turn into glycerol 3-phosphate, which can later be turned into glycerol and triglycerol and fatty acids. In this pathway, the main pathway, it turns into G3P, glyceraldehyde 3-phosphate, and it uses glyceraldehyde 3-phosphate dehydrogenase, not to be confused with glycerol 3-phosphate dehydrogenase, the glycerol and the glyceraldehyde. In this reversible reaction, it creates NADH and makes 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate uses phosphoglycerate kinase, and it produces ATP in the process. And it also turns to 3-phosphoglycerate, and it can be turned into mutase, and then 2-phosphoglycerate into enolase, and then PEP. Finally, all three of these reversible steps, it commits to the pyruvate kinase step, which is irreversible into pyruvate molecule, and this step creates ATP. So you can see and count 2 ATP from this and this, and 2 NADH, this and another one again. What I didn't show you here is that hexokinase step as well as the phosphofructokinase step each cost 1 ATP. So 1, 2 cost, and creates the ATP this entire thing twice. So 2 NADH and 4 ATP generated. So 4 minus 2, that's net 2 ATP. Next, what we have here are highlighting the 3 irreversible steps. The hexokinase trapping glucose, or the glucose kinase trapping glucose in the liver. They are the first irreversible steps because the glucose cannot escape once it's turned into G6P. The phosphofructokinase 1 is also the rate-limiting step of this entire glycolysis, this big step. That's why it's heavily regulated by phosphofructokinase 2. It's also an irreversible step. Once you get into F16P, you have to go down. And step 3 is a pyruvate kinase. Remember, kinase. So you're adding a phosphate to it, right? And it's activated by F16P. So this is a feed-forward reaction where this earlier step product induces positively mechanism forward and turn PEP into pyruvate. Now, what happens when there's no oxygen, like in aerobic respiration, to take NADH to the electron transport chain? Well, the NADH will have to convert it back to NAD+, right? In order to keep this going, or else NAD+ reserve is going to be gone. So NADH is converted back to NAD+ using a lactate dehydrogenase, and it will produce lactate as a buildup side product too. And lactate is brought by red blood cells into the liver, where it can be turned back into glucose. We also have other sugars in the pathway, such as galactose, and when galactose is made from lactose breaking down to glucose and galactose, it uses galactokinase, similar to hexokinase and glucokinase here, trap it, and a galactose 1 transferase turns it into G1P, which then turns into G6P, and so forth. Fructose is made from sucrose being broken down to glucose and fructose. It goes into the DHAP pathway, or G3P, and then they can be put back into the fructose 1,6-phosphate pathway using fructokinase as well as aldolase B. So that's pretty much it for glycolysis. So you will see that if it's anaerobic respiration, you will get acidic buildup and you get tired. Now, what we want to follow up is what pyruvate does. Pyruvate then can be taken into the mitochondria. I'm going to note it down, mitochondria. Into mitochondria, and then be turned into acetyl-CoA using pyruvate dehydrogenase, or PDH. PDH is a big complex using vitamin B or thiamine, lipoic acid, coenzyme A, FAD+, and NAD+. It's going to turn into NADH as well as carbon dioxide as a byproduct. Now, what also happens is that this is in mitochondria, right? So it basically forces the pyruvate to be transported into the membrane. Glycolysis happened in the cytoplasm. In glycogenesis, basically creates glycogen because there's already enough energy and you want to store them as reserve. This is the pathway from G6P earlier on, shown from glucose converted into G6P. It starts using glycogenin and mutase to turn it into G1P, glucose 1-phosphate, and using additional UTP molecule, it turns into a UDP glucose complex, where it can be added into glycogen using glycogen synthase as the main enzyme. Now, glycogen synthase builds it 1 to 4. Remember when we talked about carbohydrates earlier chapters? Yes, humans can only digest and build 1,4 alpha chains, glycogen. Now, when that happens, we build a long linear line 1,4, but we can also branch it to make it a lot more complex and to be more efficient in storing. Alpha 1,6 bonds are made by branching enzyme, which takes the ends and block it on like a Lego piece. To break them in the other way, to turn it back into glucose, glycogenolysis, glycogen is broken down by glycogen phosphorylase. Glycogen phosphorylase breaks it and breaks the mid bond of alpha 1,4, whereas debranching enzyme does the opposite of branching enzyme, right? Take a part of the branches and put it down into a linear path. Gluconeogenesis creates the glucose. Think about genesis, new. Neo means new, genesis, to make a new glucose. Now what happens here is when we already have the pyruvate pass in the mitochondria, it cannot be put back right away, right? So we have to kind of sneak around in an alternate pathway. Remember the 3 main parts in glycolysis that we cannot skip? Well, we could actually skip them, but not directly. For example, the first one, glucokinase and hexokinase, we have to use glucose 6-phosphatase, only found in the liver, to turn from G6P back into glucose. And here we have F6-phosphate, fructose 6-phosphate, and we saw how it's turned into F16-phosphate by phosphofructokinase 1, but it's irreversible, so we have to use fructose 1,6-bisphosphatase to turn it back. Phosphatase, you want to taste away, right? Phosphatase takes away a phosphate to turn it back. And 1,6-phosphate here is all reversible down to some couple steps. Now we have the PEP molecule. We saw how the PEP molecule is turned into pyruvate by pyruvate kinase, and we cannot turn back directly. So we have to sneak around after pyruvate is in the mitochondria. We saw how it turned into acetyl-CoA here. But instead of that, we could use pyruvate carboxylase or Biochem to turn into oxaloacetate. Oxaloacetate then can be used in a malate aspartate shuttle to transport out into the cytoplasm, and then we can use PEP carboxykinase or PEP-CK to turn it back into PEP, and we can go back this way. All right. Then finally, we have the pentose phosphate pathway, which creates NADPH as well as ribose 5-phosphate, the pentose. Now, what's their function? Well, pentose is ribose 5-pentose. You've seen it in nucleotides, so really important at building them, as well as NADPH creates 3 main things: fatty acids, cholesterol, bleach in white blood cells, as well as glutathione to protect against reactive oxygen species. So not to be confused with NADH, this is NADPH. NADH is used in the electron transport chain to generate even more ATP using oxidative phosphorylation. So how do we do this? You start from a G6P molecule. So many things start from a G6P molecule. And you turn it into 6-phosphoglycerate using G6P dehydrogenase. And then it can be turned into ribose 5-phosphate and into the pathway. Or if you want to skip this path, you could use any of the intermediates down here, such as F6-phosphate here, or G3P here, and directly use transketolase or transaldolase. You can just call them T, and they can directly just isomerize and turn into ribose 5, the 5-pentose. All right, so that's pretty much it. I managed to crunch it down. So good luck with this.